Yep.
Recording in progress. Yes, it worked. So, hi. By way of brief introduction, I'm Steve Monday. I'm part of Association of Rational Thought, which is what this meeting where Valeria is presenting. That's that group. Valeria is runs the group that you see on the screen there. Welcome to the American Atheist and Agnostic Society, and she's been kind enough to come to us to present. And the topic is brain development, fetal brain development. And Valeria's been learning quite a bit about this. I'll let Valeria, you're going to do a better job of introducing your credentials, introducing yourself than I will. So I'm going to hand it right over. And I'm going to shut off my screen because I think that lets my recording be smoother without interruptions. So welcome, Valeria, again to our group to present. This is your second time presenting to us, and we're looking forward to a third.
Hello to everyone, and thank you very much for the attention. We are honored to have a chance of presenting at your society. And just for a brief introduction, I will say that I'm a research scientist at Cincinnati Children's Hospital Medical Center. And my main focus of research currently is nephrology, but I'm also interested in many other topics and many other organs, including human brain. And the reason for that is because I think it's very important to understand how the organ responsible for human cognition and consciousness develops. And in our first presentation, and please, whenever you feel necessary, if you are interested in something or want more information, please feel free to interrupt me with questions. I'm totally comfortable with being stopped and having a small discussion as we go.
I'm going to do that right now to suggest that Julie mute her mic, because I think we're getting echo from within your room there where you both have computers turned on. Perfect.
Thank you, sweetie. Yes. So we did that. Thank you for noticing about this. So in the first presentation, which we had in the last meeting, we talked about major definition and nature and subtypes of consciousness, and about consciousness in animals versus humans. And in this meeting, I would like to focus on a different area, which is not less important. And I think I'm going to start slideshow right now from the full size. So please let me know if anyone has trouble seeing it. But currently, the question is the basics of brain development. Because as we know, consciousness requires the organ which is responsible for that, and that is the brain. And one of the papers which I'm basing this lecture on is called exactly as it stands for, the basics of brain development. It summarizes what we currently know and what is taught in the medical schools about the anatomy and function and development of the brain. And then later, in the second part of the talk, we will discuss about how brain activity can be measured, and how we can measure not just a neural activity, but neural conductivity, meaning when the conduction starts happening along the nerves. And with that, I'll begin with some basics, just some basic introduction. So human brain development starts as early as gestational week 3. And what is the important process during this is called differentiation of neural progenitors. And neural progenitors are basically like stem cells, just for the neural system. And stem cell is the cell which is responsible for the formation of the structure which gives rise to it. And there is, of course, genetic and environmental factors which play a role in this process. And both of them are important and have to play their role. So during this development, there is like a complex of dynamic and adaptive processes, and they happen throughout the course of development. And through this process, we get the new neural structures and functions. So simply from all of these neural progenitor cells, we get the full human mature brain developed with all of its neurons. And just to briefly describe what genetic factors are, so we have the genes, which are encoded in our DNA, and they represent the material substance which can be passed intergenerationally. And what we have, what actually works on the cellular level, is a product of the gene expression. And the product of the gene expression is a protein. So proteins get expressed, get first transcribed from the RNA, and then they start functioning, interacting with each other, and building structures. And usually, the particular protein can be the product of many genes, and a particular gene is one of the elements which are necessary to guide the complex process of brain development. There is a lot of genes involved in that. And just to give a heads up about how many cells mature brain contains, it's actually more than 100 billions of neurons in the mature brain. And what is interesting is that by the time of birth, the neuron-wise, it is formed. What keeps forming later in the postnatal period is the connections between the neurons. So what is a neuron? It is an information processing cell of the brain. So most important part. And here on the right, we can see the scheme of its basic cellular structure. And as we can see, it is comprised of the cellular body right in the center, and it has a nucleus. And also we see that it has many processes. Several of them are short, and they're called dendrites. And one of them is a long one, and it is called axon. So as we can guess, based upon their size, we can have both connections with nearby areas, and this is what the short processes are for. But we also can have axons, which can be as long as basically the connection path between the brain and the muscle. And they would be responsible for the conduction of signals for long distances. And as you can see, so we have about 1,000 connection of each neuron. And adult brain is estimated to have more than 60 trillion neuronal connections.
Valeria,
And also, sorry?
I was just going to say, and if I'm not mistaken, the axons of one cell reach out to the dendrites of the next. That's where the
No,
link occurs.
no, that is actually not the case. So axons of one cell would reach to another cell.
To the dendrites of another cell, yes?
Not to the dendrites, to the cellular body. They would connect the body of another cell, of another neuron.
Okay.
And that would be called
where do they connect to on this picture?
Sorry?
Where's the receptor on this picture?
Well, this is just a picture of the neuron. Like receptor by itself is a separate cell. So receptor would be localized somewhere in the periphery, for example, in your skin. And this is a picture of the neuron of the central nervous system. So there is no receptor here. It's just a representation of cellular structure of a neuron.
Wait, I mean, does the axon reach to other cells like this?
Yes, the axon would reach to other neurons. Like, for example, the neuron of the brain cortex would contact the neuron in the thalamus.
And where does it connect to inside this picture?
There is no, it's just a picture of one cell. So there is no such thing in this picture.
Okay.
If you drew the next neuron in the sequence, how would it attach to this picture?
Like, we would need to draw it, but it's not drawn here. Where I would draw it is right. Can you see my pointer?
Yes.
So here, that would be like, if we imagine that there is this long axon exceeding the size of the screen, then I would connect it here.
And each one of those little tails connects potentially to a different other neuron?
Yes, that's why we can have one neuron connecting connecting like about 1,000.
And so if on this picture of this neuron, let's say there's another neuron about to talk to it, where does it connect?
Well, I would, if they connect via the dendrites, then I would draw here another neuron with another set of dendrites, and then the body would be here. And if it's via the axon, then I would extend this axon farther away and draw another neuron right here.
Okay, I was under the firm impression that the axon's connected to the dendrites of the next in the chain, but
no, that is not the case. The whole point of axon is that you need to go to a neuron which is far distant from you. Like the dendrites are basically like, if we want to just talk to the neighbor, but the axon, the point of it is to connect, for example, the cortex with the
A
other
dendrite
cell.
of one cell could connect to dendrite of another close cell.
Yes,
I
that is
see.
correct. Yes.
Okay.
So I think the confusion, I think, is simply when the axon connects itself to another cell, no matter how far away, where does it connect on the other neuron? Does it connect through the dendrites, or does it connect through the axon, or some other way?
It connects to the body, to the
The
cellular
body.
body.
The actual, so the axon overlaps the nuclei or the cell body itself.
I
Okay.
don't think overlap is the right word. The way how cells connect is, for example, if you have the process, right, it would be a membrane, and another cell would have a membrane. So they would form synapse. Basically, their two membranes would kind of attach to each other and form channels.
I
So
see.
next-
So it's not in the picture how they
No,
connect.
no, it's not in the picture. This is
Okay.
just like, let's say if you have a kidney, we would put one epithelial cell. So that is just one cell. There is no multiple cells here.
Thank you. I'll go look up some pictures for myself later. Thank you very much.
Thank you.
Yes, or I can add more pictures how two neurons actually connect. I can do it.
That might be a nice addition to the presentation, I guess. But anyway, thank you for the answer.
Yeah, sure. I'll add it to the better version of the presentation because it's important to make sure that we can represent that. So now some basic anatomy points of the gross brain anatomy. So the evolutionary adaptation to being able to fit the larger brain size into the skull are folds, the sulci, and ridges, the gyri, because that allows you to basically fold more neural tissue inside the skull, which cannot be expanded eternally because it has to be able to pass through the female reproductive system to be able to be born. So that is why this is evolutionary adaptation for brain size increase. And so largest brain information processing networks are localized in the neocortex and subcortical nuclei. So as we can see from this picture on the right, and this is a cross-sectional slice of the brain stained with the staining, which is called, I guess, hematoxylin eosin, which allows you to see this separation. So neocortex is this 2 to 5 millimeter thick layer that lies on the brain surface. Right here, you can see the purple part. And as we can assert from the name, the subcortical nuclei are something which lies under the cortex. That's why they're called subcortical. And they would be localized in the subcortical parts of the brain, right here where I'm showing. And they would serve as a communication center. Basically, they would communicate the neocortex and the rest of the body throughout the pathways. And we will discuss these pathways later. So as we talked before, and I actually have it said here, is that the neuronal bodies connect to one another via these fibers, which are called axons, and they're long, and the dendrites, which are short. And I will for sure include Steve's criticism and include the schemes which actually show this connection, either histologically or just schematically, in the improved version of the presentation.
And
So
I got a question. In the
Yes?
in the B picture, it
Yeah.
looks like there are two halves and emptiness in the center. Is there liquid and cells in the center?
Oh yeah, we will talk about that. So there is actually liquid. You're right to pay attention to that. These structures are called ventricles, and we will have a separate slide on them.
Okay.
But yes, these are ventricles, and they are filled with liquid, mostly.
It
And,
probably bears sharing that my impression of how neurons connect came from, I'm recalling, a dentist's office floor reading a magazine when I was 8. So my information is probably about 40 years out of date.
That's okay. That's
So
actually
I'll try and
pretty
update my understanding.
Yeah, that is
I've never
okay.
bothered to question it since.
Yeah, thank you. And we can have, so multiple fibers would form bundles of these axons, and they would be named fiber tracts. And these fiber tracts, I'm just waving to my wife who's next to me right over there. And these fiber tracts, these axons, would be wrapped in the substance called myelin. And if we return back to the previous picture pretty briefly, you can see how here it's showed schematically that axons are covered with the myelin sheath. And this is extremely important because, first of all, myelin plays a protective role. Let's say if you have a damage to the axon, let's say there was a trauma and the axon's integrity got threatened. So myelin would help to recover and reconnect the axons. That is one function. And another function, which is also very important, is that myelin increases the efficiency of transmission. And how does that happen is that you see here, there's another structure called node of Ranvier. So the difference between mammals and superior reptiles and lower animals is that we have this mechanism of connection when the impulse just travels between these nodes of Ranvier, let's say from here to here to here to here, and therefore it can go very quickly, very far away. And of course, the reason for that is that as evolution happened, the sizes of bodies were increasing. And therefore, there was not enough to just have a short connection. There was a need for longer destination connection. And of course, the faster you can connect, the faster you can get the information. So that is believed to be the evolutionary mechanism for that.
May I ask a question?
Sure.
In the setting of MS, do you know where it stands for myelin regeneration? I know there's been work at it. Do you know?
Yeah, actually,
that?
mesenchymal, that's a really good question. So mesenchymal sclerosis is directly related to that. To answer, like to have an informed answer, I would need to probably read a review about where do we stand in the treatment. But basically what happens during MS is that the body develops antibodies against its own parts, and particularly myelin-producing cells. And that results in axon demyelination. And basically that results in destroying the connectivity. So was your question about like treatments?
Well, I do have MS, and the medicine that I'm on is preventing new lesions, but the atrophy in the brain volume is also a concern. And that from what I understand, the research that's into myelin regeneration will help with brain atrophy.
Oh, yes. The brain atrophy happens if you like have untreated, totally just like left it be cases. The problem is that all of these neural disorders, they for a while just weren't treated with like targeted treatments at all, only symptomatically. So we are only recently starting doing these treatments. And I think that is why there is so many cases when patients with mesenchymal sclerosis ended up like in terminal stages. But definitely myelin regeneration targeted medicines would help with that. And if that is something of the interest, I can focus on this and research about where do we stand on the anti-MS treatments currently. And
thank you very much. I didn't mean to derail this and make it personal. I was just, you started down that path and I just was very curious
there is
about
nothing
that.
personal, nothing personal at all. I'm always welcoming the ideas because that helps me to think about what could be the potential topics of interest. So that is actually very welcome. If you have a thought about a collateral question, which we could explore, please feel free to express it.
Thank you.
Oh, yes, you're welcome. And now actually directly to the question asked before. So here we have a cross-sectional sagittal slicing, which means that we cut right here and then look at it. But here we have a kind of a volumized scheme of the whole brain. And these bluish structures are actually the projection of what we had seen in the previous slide right in the center when you said that it was like a person with two arms. So this person with two arm-like structure is called ventricles. So these are the set of interconnected cavities, and they're located at the very center of the brain. And they are filled with a cerebral spinal fluid, which is recycled actually quite often, several times per day. And the function of this fluid is, of course, waste removal, cushion creation, and some transportation of hormones. So these ventricles are very important sites because during the brain development, the ventricular walls are the places where we actually have these neural progenitors, and therefore they're the sites of the neuron production. And the neurons which are formed on the walls of these ventricles travel to the cortex, and we will talk later about how exactly that happens.
Valeria, a very brief question. Is there some sort of active transport inside the ventricular system analogous to a heart, or is it all passive diffusion?
It is passive. There is no analogous of active transport because active transport involves contractility. So to our current knowledge, there is no contractile organs inside these ventricles.
Thank you.
Yeah, you're welcome. And that's a good question because I've been asked before, like the name ventricles makes the association with the heart. So that's a totally valid question. So now we're going to talk about a little bit about what the neural stem cells are. They emerge during the gastrulation, and the gastrulation is a period of embryogenesis, which is by the end of gestational week 3. And that transforms the embryo into a 3-layered structure from just first 2-layered structure and before that just the cellular association. So neural stem cells are also called NPCs or neural progenitor cells. And the reason for that is because they give rise to all the neurons of the brain and central nervous system. So they form and they differentiate, of course, under the influence of the surrounding structures. And the interesting thing is that they have the self-renewal potential. So not only they can divide and give rise to either neuron or another part of the neurosystem, which is an astrocyte or oligodendrocytes, which are playing the supportive roles. Part of them are similar to macrophages. Part of them are actually producing new myelin, as we see here on the oligodendrocytes. But these neural stem cells also have the potential for self-renewing, which means that they not only give rise to the other, more mature forms, but they can divide and renew themselves. The key thing here is that for the better or for the worse, the self-renewal potential of neuron stem cells declines over time. And the bigger the brain becomes, the more mature brain cells you form, the less of the division occurs in the neural stem cell population. And not only the division rate declines, but the apoptosis or the cell death of this population increases. And we will talk about that in our next slides. So of course, when you have such an intriguing population, there is always a void to isolate it, to get it into the cultured form and be able to culture it in the lab. Because of course, there is a goal eventually to try to induce neural self-renewing in vitro, because that can have very powerful potential for treatment of neurodegenerative disorders. Imagine if if one lost some of the neurons due to the trauma or disease, but then you can get some of the neural stem cells and differentiate more and kind of replenish for the lost ones. So there was a long effort in terms of isolating them, and there was a very remarkable paper published in 1989 when there was a successful attempt to isolation of neural stem cells from embryonic rat brains. And I would say that this is a breakthrough part, because once you have the cell population isolated, and that is actually a big part of the business, then you can culture them, you can experiment with them. And as you see, the subsequent research did growth of neural stem cells into astrocytes and neurons using the growth factors, which is another great breakthrough. And then in the subsequent phases, you can demonstrate more neurogenesis from the human cells. So that is just an outline of this portion of the research. And now a little bit of the basics about early stages of brain development. So as we said, general process starts at the gestational week 3, and then at the embryonic day 20 to 27, which is about 4 weeks after the conception, we have a structure which is called neural tube, and it is depicted here. So neural tube has anterior and posterior osteae, or they're called neuropores, and they get to close at embryonic day 25 and 27. And then the neural progenitor cells actually remain in the center of this hollow tube, which eventually all of the structures will form, and that would be the this tube space would be the neural ventricles, as you can guess. And that is the next point of the presentation, that eventually this hollow tubal space will give rise to the ventricular system of the brain. And the area where these neural progenitors will be positioned will be called as ventricular zone, or VZ. And logically, we can infer that the most anteriorly or frontally positioned progenitors will give rise to the frontal parts of the brain, and then the posterior parts to the hindbrain and to the spinal cord, which is actually located in the spine, and which connects the cranial brain to the periphery. So, and now, so once we have that, once we have the brain, we have the cerebellum, we have the hindbrain parts, and then we have the spinal cord. The very important question comes about how is it all connected to each other, because this is large. And here we come to the structures called the pathways. Pathways which actually are comprised of these neural bodies located in the center or in the periphery. And these axonal bundles, which connect one neural body to another neural body localized in a different brain structure, or in the spinal cord, or if we go further and imagining the scheme further, it would be down in the spinal cord, and then we would go to the peripheral muscle. And there are these two crucial pathways. And why these two pathways are particularly important is because basically, without them being formed, there is no connection between the central part of the nervous system and the rest of the body. And these two pathways are called thalamocortical and corticothalamic. And their formation is complete, just histological formation on the histological level, is complete by the gestational week 26. And as we can infer from the name, the thalamocortical pathways connects this structure called thalamus. And thalamus is a very important brain element. So when I'm explaining what are its functions, I always compare it to, like, for example, if you have an underground train station, or if you have a bus station, then there would be something in the center which would guide the directions and which would receive the input and then give the feedback from multiple places, and then eventually guide where everything should go. So the thalamus is kind of a coordination center of all the sensory and motor information. And as we can infer from the name, thalamocortical pathways would connect first the peripheral receptors to the neurons located in the thalamus. So if we think about this picture, which I had in one of my first slides, just imagine that this central structure called cell body is located somewhere in the thalamus. And then we have an axon, which would go to the cortex and form another synapse in there with a cortical neuron. And corticothalamic pathways, again, as we can infer from the name, stands for the opposite direction. So let's say that the cortical neuron wants to talk to the periphery. So there will be a cellular body here, and then there would be the axon, which would go to thalamus, where we'll have another cellular body. And then this thalamic cellular body would give out an axon, which would go into the spinal cord and go through the spinal cord to the level where it's intended to be. Let's say if it wants to connect to the arm, it would be on the level of the arm, or if it will go lower to connect to the muscles of the lower extremity. So basically it would connect the cortical neurons through thalamic neurons and then spinal cord to the periphery. And again, the very important time point in the embryogenesis gestational week 24. By this time, these pathways are formed. And maybe to kind of imagine that, we could think about connecting what we usually do when we start our meeting in the library. We always have to connect the monitor to our computer to be able to stream our presentations. And basically these pathways function as a cord without which there would be no connection of the cortex to the periphery. And this is, as we currently see, absolutely required for the most primitive forms of consciousness. Because before that, there is no possibility how the fetus can, even on the purely physiological level, control the rest of the body. First of all, the thalamus forms between gestational week 24 and gestational week 28. So without the thalamus, of course, by itself, it's not possible, but even without the pathways, definitely.
Valeria?
Yes?
So the neurons that are going down the spine, is there usually at least one more connection where like the nerve in my fingertip reaches to the spine neuron, and then is there another junction within the spine that connects to another set of nerves that reach out to the periphery?
Yes. So as you can see here, there is an attempt to depict that. So you
Well,
see
I'm talking about
this...
below that. I'm saying, doesn't the green terminate in the spine, and then there's another nerve that reaches out to some part of the body, or does that
yes,
reach out
definitely. So
Okay.
there would be another nerve. Like if we had a full picture, then we would have another nerve, which would, for example, go out of the spinal cord through the spaces in the bones. And then this nerve would actually go through the arm or through the leg to the muscle.
So it is nominally 4 axons to get from the cortex to the fingertip.
Yeah, so there will be one axon from cortex to the thalamus, another axon from thalamus to the spinal cord, and then another axon from the spinal cord to the periphery, so 3. And 3 neuronal bodies. So there will be one body in the cortex, another body in the thalamus, then another body in the spinal cord, and then it would go to the muscle. And of course, muscles will also be connected to this axon, but it would already be not neuron, it would be a muscular tissue. And
Steve,
if we had...
Are you asking how long an axon can be? Because I think the answer is tens of centimeters.
That's an adjacent question. Yes, I was actually trying to understand whether it was 3 or 4 to get to the fingertip, essentially.
So it was 3. In this pathway, it would be.
So you're saying
3.
that green neuron goes down the spine and all the way out to the muscle or out to the sensory?
No, so this green would go down and then form another axon right at the level of where it form another synapse. Sorry, form another synapse, and then from that synapse we would have another axon, which would go.
That's what I thought. So 4 total. Yellow, white, green, and one that goes out to the periphery.
No, so if there's just like a picture. So if you think about that, you would have this axon which can get out and go to the periphery.
So
it's just like not in the scheme.
Okay, so that means tens of centimeters.
Yes, it can be quite long. It can be tens of centimeters.
But 3 connections.
Yeah, one axon can be quite long.
Okay.
Definitely can be tens of centimeters.
Let me ask the mass question.
Yes.
What are the shortest axions, and what are the longest axions? The max and min they can be.
That one I would have to look it up. I can check this information and then write it in the comments for the meetup, because I don't want to just eyeball it. Okay. I want to get the precise number.
Okay.
But it can be pretty long, because like that is the whole point, that you can connect these to the periphery and that you will form another synapse on the level, for example, of the leg, which means that it would be as long as from thalamus to the leg. So I will look it up for you and post it.
Thank you.
You're welcome.
To show a little of the magic of the system, Steve, I think you're getting at something very fundamental. All this marvelous information processing and sensing has occurred through a small number of layers of cells.
Yeah. Well,
Definitely.
it at least makes its way to the cortex in a small enough quickly because
Yeah.
it's a small number of cells.
All right, and now we move to more detailed information about the cortical organization. So we went through this on the level of gross anatomy when we were talking here, but now we're actually going to go deeper into the histology and talk about exactly what are the cortical layers. And as you can see here from the pictures A and B, so this is the direction of the cut, and here you have it magnified. So we will go from the bottom level, and as you can guess from this shape and from the localization, so this resides on one of the outer walls of the brain ventricle, which we talked about. So here we would have first the area called ventricular zone. And as we already discussed in our previous slides, ventricular zone is where the neural progenitors are located. So this is a very important zone, and due to its high cellularity, it will have high MRI signal intensity. Then above it, we would have the subventricular zone, and you can see this literal F, fibrous. So it would mostly have fibers, and that's why MRI intensity would be low. And then we have the part of the subventricular zone cellular. It's not depicted here, but it would be above the fibrous, which is cellular rich zone, and it would have moderate MRI intensity. And then we would have the intermediate zone right here, which you can even see on the histology. And then we would have the subplate zone, which again would have a lot of fibers and a lot of traveling cells. And as a result of that, we would have low MRI intensity. And then the final, we would have the cortical plate, CP, where the cortical neuronal bodies reside. And due to that, you would have high MRI intensity. And the MRI image is actually located on the C on the right. So this is just about the structure, and I will explain why this is particularly important later. So it is important because through all of these layers, as you can guess, the forming neurons would be traveling. Like, let's say if the neuron progenitors are here, they differentiate, they divide, and then they would travel through all of these levels to finally result in the cortical plate zone.
So MRI signal intensity is an indicator of high metabolic?
Of high cellularity. So cells and fibers would have different histological elements and different densities. So due to these different densities, there would be different MRI signal. But no, MRI cannot detect metabolism. To detect metabolic activity, you would either need to like take the postmortem sample and do the staining, or there would be other methods which would like detect the ion currents, for example. And we'll talk about some of that, but I don't think it's MRI. So as you can see from the structure, the subplate zone is actually thick, and it represents this area where these fibers from the, let's say if we have the thalamus somewhere beneath the ventricles, then the fibers from the thalamus would go through all of these structures up to here, and here they would undergo arborization. And what are the arborization? We briefly touched this. So let's say when you have your extremity, then you have this multiple smaller processes which allow to connect with more neurons than one, and that would be called arborization. So once the axon grows, then it eventually undergoes arborization. And then the goal of that would be to connect to the subplate neurons. And this process is extremely important because when the researchers were questioning, well, what is the role of these subplate neurons? Are they even needed? And maybe they are not needed, and maybe all what has to happen is the connection between thalamic neurons and the cortical plate neurons. They've done the deletion of these subplate neurons using the transgenic mice. And of course, when you do that, nothing happens. So there is no further stage of development. There is no formation of cortical plate neurons. And from that, they inferred that the subplate neurons probably have a crucial role in guiding the thalamocortical fibers to their eventual target, because without them, this doesn't happen. So now the question about
Valeria?
how can, sorry?
I'm sorry, I just want to make sure I understood that. So
Yeah.
the subplate neurons, they are neurons.
Yes, they are neurons.
They are neurons, but after growth has occurred, after maturation has occurred, you can remove them and there's no consequence. So they're not
And actually,
involved in
that
the
is exactly what
connectivity.
happens. You probably remember from our presentation that there are things called regressive events in the brain development. And actually,
right.
yes, you are absolutely right. The subplate neurons, after they've served their purpose, they undergo what is called the programmed cellular death or apoptosis. So they're one of the things which are removed. But
Which
if you remove them before, then there would be no further stages.
Which you'll come to, I guess, in a few slides.
Yes, we'll come to that. So
Okay.
the question here is, how can we examine all of that? So definitely from what had already been presented, histology is a very helpful tool because it allows you to detect changes in vitro, and you can do not only like electronic microscopy like here, but you can also do fluorescent microscopy. And what it would be is, for example, you can stain with an antibody against a marker of, let's say, cortical plate neurons. And then you can treat it with a secondary antibody, which is against a species in which the primary antibody is raised. And that secondary antibody can have a fluorescent tag. And that would make you capable of imaging this whole structure using the fluorescent microscope. And that is, I do a lot for my actual project. I do a lot of the IF immunofluorescent stainings, but so far not only on the brain, but mostly on the kidney. So,
can I ask one more question?
Yes.
So so before this gestational week 21 to 23, the connections between the cortical plate and below don't exist yet?
Yes, because they first have to, like, you first have to get enough neurons, then you have to get enough fibers from the thalamus. So before that picture, they are not detectable, and we will talk about that, how using histology, they can see at different stages of brain when the connection is formed and when the neuronal body is formed.
And the interconnection between cortical neurons is also forming during this time?
Interconnections form later. That is the thing.
Later still.
Yes, they do form later. And that is actually not phenomena, but just a critical feature of the human development, is that by the time of birth, number-wise, neurons are formed. We have about the same number of neurons we'll have throughout the whole life. However, the connections between the neurons are what keeps forming and forming. And actually, it goes quite long into the development, because the full connection capacity of a fully mature brain is reached by like the third decade, and particularly in the frontal parts of the cortex, which are responsible for executive function. So connections keep forming much later in the day. Definitely, another method would be MRI, and that makes life in vitro a little bit easier because you don't need to extract the tissues of the either post-mortem or of an aborted fetus to study that. That can be studied in the living person. And I found another name for another method in this review, which I read, and it was called Advanced Diffusion Tensor Imaging and Automatic Segmentation Algorithms. And that actually what was done to study this cortical lamination. So you would do these two methods, one in vitro, when you, let's say, extract the sample of an aborted or stillborn fetus and just do the electronic microscopy or stain. And another thing would be this, and that would allow you to do these studies in vivo without having to kill the thing.
Valeria, could
Yes?
you give a quick notion of what you mean by histology? It sounds like a method.
Yes, so what is histology is you take the organ, let's say you kill the mouse, take out the brain, then you do special procedures to fix it, which means prevent its proteins and genes like RNA, DNA sequences inside it from degeneration. And that is done with some fixatives. We can use paraformaldehyde. That's what I'm using, paraformaldehyde, a lot. Other people use formalin. Some people use methanol. So there is several chemicals which are known to preserve the organ in the shape it was right after the extraction. So you do that. Then what happens is you put this organ into the VAX. And what it is for is that once it's placed in the VAX, you can section it into very tiny structures on the microtome. And actually I'm lucky because that's exactly what I've been doing yesterday. And I have taken some pictures for that. I can't show you on the slides, but can you see what is on my cell phone?
Well enough.
So this is the microtome. And what it is that you see this central 2 red kidneys in a piece of white wax. So you put it like this, and then you slice it. And the wax allows you to slice them very thinly, like you can do
like a deli slicer.
5 microns, 4 microns. So there's like a lot of different thicknesses you can have. And the ultimate result looks like that. Do you see these like kidneys on the slides, sections on the slides layered out for drying?
Yep.
Yeah, so that is exactly what histology is, and that's what I've done yesterday. And I have to say thank you to my wife, Julie, for picking me up from work at what hour? I think 11 p.m. I think it was around 11 p.m. So yesterday I've done a lot of preparation for histology. And I'm glad that you asked this and I was able to show you directly. And basically, once you have the section, you can do whatever you want with them. You can do some electronic microscopy, which will result in the image like this. You can do the staining for what I discussed, like, for example, you're interested in the protein, which is a marker of cortical plate neurons. Then you buy an antibody, which is against that protein. You do the staining protocol, you treat the section with this antibody, then you treat it with secondary antibody, which is fluorescently tagged, and then you image.
Now, when you get your information, is it always visual or do you test them chemically?
You can test them chemically. Actually, that would be a good metabolic study. So there was a question about metabolism before. You can treat them with certain chemicals, and that would show you which metabolites are present in the certain layer of the brain. That is one thing. You can do it chemically. Another way would be you would just do antibody stain, which actually is immunohistochemistry or immunofluorescent staining, depending on your methods of secondary detection. If your secondary detection is based on the chemical reaction, then it is called immunohistochemistry. If your detection is based on the fluorescently tagged secondary antibodies, then it would be immunofluorescence.
Okay, now is all this information like a snapshot in time, or is there some mechanisms that continue in the tissue that you can study in terms of change?
To study in terms of change, you need to collect at multiple time points, because there is no way how you can do this. Just like, if once you've taken the brain, it is over.
It's over.
Apparently, there is no continuation. So when I actually do a lot of these studies, like, for example, when we ask the question how the kidney disease evolves, I would do surgery in the mice and then collect samples at, let's say, day 1, day 7, day 28, several months. So that would be what is done for studying the process as a function of time. Mm-hmm. For those who work with things like zebrafish, for example, or insects or plants, or if you just work in the cell culture, what you can do is a direct live imaging. But the problem with that is that you can't do this for the brain, because we are not at the point of research yet when you can just grow a mini brain in a Petri dish. But once we are there, and I believe that we will be there, then we can do in vivo imaging, which means that you can simply, let's say if your brain is grown with a built-in fluorescent reporter, and that fluorescent reporter makes your cortical neurons all green, then you can technically track and do the in vivo timing experiment from day 1 to whatever day you want. And you wouldn't need to kill the thing for this, because all you do is you take your Petri dish to the fluorescent microscope, which has a capacity to image through all of these cell layers, and you do that study. And people who work with zebrafish and who work with insects, they do a lot of that.
Thank you.
Yeah, but I can't do that because my model is large enough, and there is no way how you can just image the kidney on this resolution without actually extracting it. So if I want to ask a question of time, I just do multiple collections at multiple time points. And it is very important. So this was just the previous slides were showing more details and at the previous stages, but now we look at the later stages, and we just have a have a collection of main events, just a summary. So main events of the previous stages would be that we generate large numbers of new neurons in the ventricular and subventricular zone. So you have to get to the numbers of neurons before. And then we briefly touched this topic. The neurons start migrating, and they would migrate from here. You see this little cavity here, the white part, that would be the ventricle. So the ventricular zone would be right above, and it is here. And the neurons would be migrating all the way up, and eventually ending up in the cortical plate. And those are the fibers from the thalamocortical pathway, their afferents, they would be growing and they would be accumulating in the subplate zone. And the major event of this early preterm phase is when the thalamocortical axons would make a connection with the cortical plate neurons. That is when your pathway is complete, because as it comes from the name, if you have a thalamocortical pathway, the moment when the thalamic afferents connects with the cortical neuronal body, that is the moment when you can start the conduction. How we can examine this? So one way would be histological examination. And what you do for histological examination, you connect your organ at multiple stages. Let's say you want to connect brain at the week 15, at week 20, at week 24, and then you slice them in the way I have shown, and then you look at that and you see what are the structures which are necessary for example, connecting between the two.
You're actually counting up new synapses.
Yes, exactly. And this is the electronic microscopy of exactly what you said, a synapse. That is how a synapse looks on the electronic microscopy. And you can detect these structures, and once you see that these structures are formed, then you can say that your synapses are present in there. But that is a morphological way of detecting things. The next question is how...
I was just going to say, one of the big questions and discussions that came up when she did these presentations in the other group, mostly for me, I was trying to understand, when can consciousness begin? And so if you understand consciousness to be a result of internetworked neurons, then it ain't happening between about gestational week 23, because you don't have the synapses yet. There's no way to have a networked anything. So that was a very interesting point to me, is, at what point in this process is it possible for consciousness to exist?
Yes, that was a really good question. And then, so there is always, not only in this field, but in every field, like morphology, histology just shows you that the structure is present. But how to know if the structure actually works? And that would be answered in the next slide. But before that, I would like to summarize just the results. So once you have the thalamocortical afferents in growth and synaptogenesis in the cortical plate, what does that give you as a summary point? So you would have connection with the sensory motor cortex, which would be located right here. You would have connection with the visual cortex, and the visual cortex is located in the posterior parts of the brain. You would have a connections with auditory cortex, which is kind of around your ears in the parietal parts of your cortical lobes, and that would be about here. And you would have connections with the frontal and prefrontal cortex. So all of that would be here. You see this green structure. So these are where the thalamus is depicted. It is ventral anterior and ventrolateral nuclei of thalamus. And also at this point, you would have the pain mediating pathways become available, because pain processing is also related to the formation of these pathways, and is also takes place in the thalamus. So these are just the outcomes. But aside from histology, like, okay, let's say that we know that synapse is present, but how do we know it actually works? How can we detect that the conduction, and what this conduction stands for, is the process of transmitting the signal from the neuron to another neuron? Because as we know, neurous tissue is a tissue capable of conductivity, which means that it can transmit these electrochemical signal from one part to another. So how can we know that this synapse actually does work? And I read several papers about that. And these studies are done, of course, with all the appropriate consents and processes in the preterm born babies. Let's say if you have a baby born at the gestational week 22 or 23, 24, 24 is usually the time since which they can survive independently. So you can do this stuff, and there were institutions which did these studies. And that is called full band electroencephalography, or FBEEG. And the principle of it is quite similar to the principles of studies of the cardiac rhythm. When you, for example, in patients who have arrhythmias or poor cardiac conductivity, you would put them on the electrocardiography, and they would also have the same electrodes placed around their heart. And through that, you can see if there is something called action potential. And what is action potential is exactly the sign that the conductivity happens. If there is a conduction, then there is an action potential. If there is no conduction, then there is no action potential. And the good question which was asked in our previous meeting was like, how precise is that? Because like people move a lot, especially if they're babies, and like, how can we locate from which layer does the zone come? But the good thing to remember here is that we're talking about really high numbers. So we have, by this time, more than 100 billions of neurons. This is more than we have cells in the heart. So if it works in the heart, and if we can locate from the electrocardiogram in which part of the heart there is an infarct, and it has less cells than that, then we definitely can do it with these numbers, because there is the neurons, remember, are formed in terms of numbers by the time of birth. It's the connections which form later, but in terms of numbers, they are there. So that is the method which I was able to find to study this neural conductivity. And there would be another one. So that is how it looks if you try to map your action potential to the scheme of the histology of your cortex. So you see how there are these different action potentials drawn here. So this is just an early preterm. And if you compare it to the scheme before, you see that it doesn't even go to the full phase. And the reason for that is because at this stage, there are only individual thalamocortical axons right here in the subplate connecting to the cortical plate neurons, which are shown here as a scheme. So since there is just a few of them at the early preterm stage, which is gestational week 23, 24, you can only have insufficient, incomplete action potential. You only have the phase which is called long depolarization. But then when you go later, at gestational week 25, then we see that there is more synapses formed. And
Larry, is this single cell recording?
no, this is not single cell recording, but this is a layer recording. Like we can detect from which layer the signal is coming, and we can detect like what is the form of this action potential. So it doesn't have to be single cell. It's like in the heart, like as long as you on the electrocardiogram don't see that the action potential from this part forms, that's how you know that there is likely an infarction in there.
You're
So
trying to generate some.
This thing doesn't have to be single cell. You do this study at these times, gestational week 23, 24, 25, and then you match these results with the histological samples of, let's say, stillborn or aborted fetuses. And then you match the structural data to the function. Let's say you detect that at certain stage there is like very few synapses in the subplate. Then at the week 25 you have more synapses, and then later you have the full amount of synapses. That's your morphological part.
I think I understand that. That's very good, but you're detecting whether or not a lot of cells are giving you action potentials via a gross, response, I think. But it seems to me that requires you sending a single well-defined signal from to the neural plate. I don't know how you do that.
But I didn't get, like, why does it have to be single cell?
You're saying this is not single cell. Those recordings are not recordings from single cell electrophysiology. You're saying that this is a gross anatomical response. It seems like there must have been a certain stimulus for each one of those potentials in order for you to see response. I'm asking what that stimulus was.
But there would be no, I don't think there needs to be a stimulus, because it's like the heart is beating normally in a living organism.
Well, but the heart is a rhythmic organ, the brain isn't.
Yes, but it's not about rhythmicity, it's about conductivity. Not all conductivity is definitely connected to contraction. It's like, there is currently no direct stimulus in my brain, but my neurons are still generating action potential. So
why wouldn't you see continuous noise?
Sorry?
Valeria, what David's trying to say is that the heart synchronizes the signal. Here, what you're showing is in the patterns are the fact that all of these interactions are synchronized. And as they're synchronized, you can see an overall potential, which shows partially the full synaptic behavior and the depolarization behavior. The question David's asking is, why do you get a synchronized signal coming out of these plates?
Well summarized.
Why? Just because once the structures form, they start functioning.
So they self-synchronize, is what you're trying to say.
Yes, like once you have, that is the whole point. Like once you have a synapse, you start conducting. And at this point, to detect the conduction and action potential, you wouldn't need a special stimulation.
So it's not random static of individual neurons. There are waves of electric activity.
Yes, exactly, exactly. And once you can detect these waves, then you know that your synapses not only just form but function.
So they're local in a very localized area. There's a wave of electrical activity.
And to localize that, you position the electrodes there. So you can of course do like a stimulus study, but that would be a different question. And I think we partially touched that in my later slides. So
from a layperson perspective, it sounds to me like what you're saying is, in some sense, the organs themselves take over control and the brain is a response to it rather than the other way.
Yes, 100%. Like that's what we were talking about in our earlier parts, is that a lot of this happens under the control of genetic, but also environmental factors. And how the environmental factors play a role here. For example, they would guide the migration. Like there would be a thing called paracrine signaling. And what it is that once you have a certain structures, they start producing certain chemical, certain molecular signaling to surrounding cells.
environmental stimuli starts the processes inside the body.
Yes,
to
exactly,
produce
exactly. Like
signaling.
this, if this process wasn't self-regulatory, then it wouldn't be even happening. Because then we would need like some sort of external force which would be guiding this process. But the thing is that it doesn't have to be, because it just occurs.
So that
And what
involves the assumption that a small region of cortex responds to some kind of stimulus synchronously somewhere else. So there must be a region that's small enough to do that, but not so large that more than one function is involved.
Yes, I would
Yeah.
agree with that. And what it is believed to be is, for example, that there are paracrine signals from these cortical plate neurons, which tells these axons to grow there. And remember, we also talked about subplate neurons. So there is another explanation for that, that the subplate neurons produce substances which also guide these axons to grow towards a certain direction. And that is not only how it works in the brain. In the kidney, it works in a very similar fashion. When we did, actually in the kidney, we were able to do it on the single cellular level because we did single cellular sequencing of the individual renal cells in both health and disease. And
So
then
in a way, you're using collective firing to tell you that synapses are operating.
yes,
Okay.
yes, that
So-
is what is done at this stage.
I would remind David that people have talked a lot about when taking electroencephalographs, and you see varieties of waveforms, which are actually, of course, collective behaviors of the brain cells.
In adult brain, sure.
In adult brain. So I'm just saying that all we're saying is that the same sorts of behavior is apparently there as well in prenatal even.
Yes, absolutely right. Yes, that is exactly the case.
At
And
the risk of taking us down another segue, the last time you went through this, on that previous slide, you keep emphasizing the completion of it coming back down at the end, and I didn't understand why that's significant.
what do you mean coming
The
down?
little spark line at the top,
Because
you're
that is the
emphasizing the
full.
full. You keep saying the full potential, and I don't really understand what the importance of that little curve at the top coming back down indicating the completion of something is. All I'm understanding. I don't understand what it's the completion of, I guess.
Of the single action potential. So action potential in all electrically active tissues has phases. If let's say
Okay.
the action potential can go only up to here, that means that there is no full conduction. You can do the same studies with the forming heart or with a heart which is infarcted.
It
And then
means
you would
they're
see
not.
exactly.
It means there's not a full connection.
Yes, that means that there is not full connection, and that the reason for that is either there's no synapses yet or not enough synapses. And then once you have all of them formed, then you have the full action potential, because then you can have the full connection from the thalamocortical fibers to the cortical neurons.
So the charge is building up, but unless it goes somewhere.
Steve,
Maybe
I think she's
maybe
talking
that's the way to
about
think about it.
number of synapses, not the behavior of a single synapse. Maybe observe that your previous slide talks in terms of action potentials, which are usually described at the single cell physiology level. It's not so much that you're looking at single action potentials in this case, you're just looking at synchronized firing of individual neurons. The ones that are active have their own individual action potentials. But the response of a piece of tissue is, I think the awkward thing is you say action potential, and action potential is measured first in an individual terminal. I buy the principle you're outlining. You're really looking at what the number of functional synapses are with this technique. That I buy. It's just that the terminology threw us off, I think, or threw me off at least.
Yeah,
Should
I can correct that and look at how to better describe that, because that's how it was described in the paper. But
should I
will.
sorry should I think of it as that first little spark line that just goes up? You're building up a potential, but it's not able to go anywhere. And by the time you get to the picture
I
on
would
the
agree with
right,
that.
That
I
you've
would
built
agree
up the
with that
potential and it's dissipated because it's gone on down the line. Is that the way to think of it?
I think yes. I would think so. But I will look more about that because like this is the terminology which was used in that paper.
Fair
But I
enough.
look at
Yeah,
other
I just
papers.
I know I didn't get it. I knew that you were never mind. I won't belabor it. I think you've I have a deeper understanding than I did last time, and if I see you do it a third time, maybe I'll get a little better.
Okay, and I can add even more information. I'm actually curious because maybe since that time, since 2006, they very well might have done the single cell physiology by now. So that's a good question. I'm going to look it up and what was the progress in this, because this phase is heavily developing. So they might have been advanced at that as well. So I will look up more of this information. And now coming to what I would like to describe as non-pathologic regressive event, which happened during brain development. Most of things we were talking about before could be classified as positive events, progressive events, when we generate new structures from the cells. But there are also some, surprisingly, completely non-pathologic, normal regressive events, which means that structures which form die. And the fascinating fact is that up to 50% of the formed neurons prenatally die. And not only that, but they die in a process which is called a built-in programmed cell death, known as apoptosis. So we know that there is two major forms of cell death. One is necrosis. And necrosis is a cell death which is not programmed, not built in, and it is pathologic. It results from either injury or lack of nutrients. But this form of death is a programmed form because it has pathways which just are encoded in the DNA and which get activated, usually in response to some external stimulus. And the fascinating thing is that in certain areas, and one of these areas is actually a subplate, which we talked before, so the SP subplate cells. In these areas, apoptosis rate can reach up to 70%. And the way how we detect it is using histology. So you can take the specimen, stain it for the apoptosis markers, which are very well known and well studied, and then you can simply quantify what is the percentage of apoptosis markers positivity in a certain area. And the apoptosis happens via nuclear chromatin fragmentation and then eventually cell breakdown. And what is particularly fascinating is that there were several hypotheses about how this happens and what guides, what triggers these processes. And we know that there are certain neurotrophic substances which are generated throughout the brain development and adulthood, and that these substances are protective against apoptosis. And there was one of these hypotheses generated in 1989 by Oppenheim, and it was called neurotrophic hypothesis. And what it was standing for is that the neurons which have more connections are capable of obtaining more of this neurotrophic substance. And the substances can also travel in the special transporting vehicles, either through dendrites or through axons. They can travel long destination, short destination. So the hypothesis stands for that the neurons which establish more connections are capable of survival at higher rates than the single ones. And the way you can test this is that you can, again, do the histology. Let's say you do staining for the apoptosis markers and for the neuron markers. And then you can simply calculate where the apoptosis rates are higher in the neurons which are well connected or in the neurons which are single. And the data points behind this hypothesis is that the apoptosis rates were higher in the disconnected parts than in the connected parts. And then the next step is that you can stain for these actual neurotrophic substances because they're all proteins and you can develop antibodies against them. And then you can stain for them as well and see if the apoptosis markers colocalize with the neurotrophic markers or not. And an important point to mention here is that among the transient populations, such as, for example, these subplate cells, the apoptosis rates are higher, and which touched this point before, among the neural progenitors, the apoptosis rate is also the highest. Not only that, but these rates increase as corticogenesis occurs, which means that the rule of brain development is that the more mature cells you have, the faster depletion of the progenitor pool you have. And that is an interesting point.
So Larry, I've got a kind of a wild ass question. So it seems to me that and I don't know any real detail, but I've read that the brain in its early phases of evolution, or not the brain, the whole organism in its early phases of evolution tends to go through various earlier almost evolutionary phases as it is developing. Shows signs of different, more or less animalistic phases in development.
Yes.
And I would wonder if this is the brain equivalent, and that, in other words, the neurotropic substance actually is the developing factor which creates our modern brains, and that what you're seeing actually are developments of earlier brain formation. So if you did comparative anatomy on the brain formation or the brain structures of earlier animals, would you see some of the structures that are evident early in the development here, which then go away here, they disappear here as we continue to develop the brain into a modern human organism, but which actually correlate with the structures you might have seen in some of the brains of earlier animals?
To a certain point, there is a point of divergence. So there are points which are earlier, which are very similar among the mammals as a species. But then when you come to the development of the cortex, that's when the differences start, because the human primates and humans have much larger cortices than, for example,
right.
a hedgehog. Not
right,
only
but does the programmed death represent the loss of structures that are no longer necessary that were valuable in earlier mammals, let's say?
I got the
Actually,
impression
no,
like
these kind of regressive events are not observed. That is the interesting thing, because in the kidney, that is exactly what happens. For the kidney, you are 100% accurate. There is pronephros, which is like in amphibians. Then there is mesonephros, which is like in the reptiles. And then there is metanephros, which is in mammals. And in that development, this apoptosis helps to get rid of the structures which were important for the previous species. So some of them die off, some of them just stop growing. But to my knowledge, not in the brain. Like in the brain, apoptosis is not targeted to destroying something which was important for the previous species. It is targeted to removing the transient populations and the progenitor pools. But it's not that we form some structures which are specific for primates, but then they get then they get removed by the programmed death.
At least the bits that you're describing are not structures that are dying off, but
layers.
Well, yeah, they're trying to searching for the right words. They are the SP cells form to get a job done. When the job's done, then they die off.
Yes, and they die off. But what
cells
I
are formed to get a job done, and when the job is done, they die off. It doesn't seem like
yes, but I thought that the question, if I understood it correctly,
yeah, it was
a different thing, because, like, for example, pronephros has
you
structures.
did understand the question.
Yeah, so pronephros has structures which
recapitulates philology, I think was the original form, or something like that.
Yeah, my only additional point was that these two examples she's describing here in the brain don't sound like that.
No, it doesn't sound
they
like
do
that.
not. They do not sound like that, because even in the reproductive system, like the basic form of development is the female organ, for example, the Mullerian duct. But even there, the Mullerian duct doesn't die. It just starts, it doesn't keep growing, because certain genes on the Y chromosome get activated And instead of it, there are other structures which form, but the Mullerian duct remains rudimentary. So it's not completely being removed. It just stops growing at a certain point.
Yeah, the fancy term was ontogeny, recapitulates phylogeny, which is not
Yes, yes, we
apparently
studied
not
that.
what's
We
happening
studied that
here.
all in biology.
No,
Yeah, really
apparently
sweet
we're
good
back in the
times.
18th century. You know,
How-
Yeah, sweet
poetic.
good times. You're giving me, how is it called, deja vu? Like I remember the
Deja
school
vu.
classroom when we first learned that phrase.
Talk about
Yeah, that's
deja
just
vu. When I did my graduate studies, I was reading Katz's original papers establishing that synaptic transmission occurred via exocytosis. So your EMs are a trip down memory lane for me.
yeah, I understand that.
All right, I have a question which I think
Sure.
is a naive version of what's being discussed, because some of this is over my head. But here's my question. If you, is there research studies when you tweak apoptosis, if you're able to tweak it, can you do things like increase IQ? Or, certain, is that a way to change development and create almost advanced species?
I
Is there-
haven't seen these studies, and I wouldn't think that they would help because it would be about connections. So that is what is, I think, more contributing to the IQ, because the numbers of neurons don't vary that much between the humans, but what it is the connections, and that is actually something which which learning is believed to be involved with. And by birth, we have our almost ultimate amount of neurons, but not amount of the connections. And even
Yeah,
as my
but
second,
does apoptosis, can that be used to create more connections or less?
I think it's a different kind of cell that's dying off.
exactly, yeah, that's a
Oh.
different process. So apoptosis is targeted at killing the cell.
Oh,
To prevent the connection, you would need to disrupt the axon. This is more of a reminiscent of what happens in the mesenchymal sclerosis. When the
Okay.
neurons by themselves don't die, at least not from the very beginning, it's the axons which are destroyed.
oh,
Yeah, and I hadn't seen these kind of studies, but I can look up what happens if you tweak the apoptosis. But I actually think that according to the logic of this development, it might very well ruin the brain, because all of these cells probably are connected to each other via the paracrine signaling. And what might happen is if you prevent the SP cells from dying, I would predict that it actually might lead to malformations. It would be like, for example, non-closure of these anterior and posterior osteae when you have a certain forms of meningocelia, or when you have when you have like the fluid structures in the brain. Like
well,
I would think
what
that
would
this
your
would lead to
prediction be the other
pathology.
way? If you made them die sooner, has that been tweaked?
Yes, that is what I talked. So when you try to make them die sooner, the further development doesn't happen. Like you wouldn't have the cortical neurons migrating properly, and you wouldn't have them connecting with the thalamocortical pathway. So if
So
you
it's somehow evolutionarily optimal.
yes, definitely. I mean, as everything. So this
Oh.
timing is evolutionary optimal, because if it happens too early, then you don't have the job done. And I would think that if that doesn't happen, my hypothesis is that it would lead to pathology, because certain populations have to disappear. If they don't disappear, then they would form the structures which are normally not present. So I don't think that this is the way to tweak IQ.
Okay.
In people. I think that a lot of promises will come from our capacity to regenerate the neurons which were destroyed by the injury. And I know that there is a progress on that. A couple years ago, I've seen that scientists in Israel managed to regenerate the spinal cord neurons after the traumatic injury, and that resulted basically in return of the function. So I think that the direction we should go is we should try to learn how to regenerate the lost connections and lost neurons. Like, for example, if someone had a brain stroke and lost part of their brain, we should try to learn how to make the surrounding neurons to either take up the lost functions or regenerate the lost neurons.
Valeria, so Alice is asking in the chat here, and you've kind of partially answered it at least. She asks, what if apoptosis misses some cells and they make it to the phases into after birth? And what so you kind of answered that. You thought some sort of pathology might happen, and then she followed up with what type of pathology could happen hypothetically. Do you have any sense of that?
Yes, I have a sense of that. I think that what would happen is similar to when, for example, the brain starts developing not in the skull, but in other parts. So that could be one direction. The other direction could be that this whole pathway would get kind of messed up, and instead of having synapses with the cortical neurons, you would have synapses all over the places, and that would result in basically signal not reaching the goal. That would be my other hypothesis. And another thing could be that since it is programmed that up to 50% of formed neurons die, if too many neurons stay, maybe as a result, each of the neurons would have less space for development. Like if there is twice as much of the neurons, then each of the neurons would have less nutrients, less blood supply. So the eventual outcome could be that there is less, there's like ischemia coming from more cells which need to be fed.
It ain't good.
It is not good. No, it would likely, yeah, this needs to be tested, but these are my propositions. And I can again look up this if that had been done in the rodents, because apparently you can't do this in the humans. But in the rodents, they might have tried to like block the apoptosis and see what would happen.
So Valeria, this is a little off topic, maybe, but this raises a kind of an interesting way of looking at this whole discussion that's had about nature versus nurture.
Mm-hmm.
And in that, in an ideal world, so clearly the brain is plastic, especially during early development, those 30 years, in fact. So therefore, and you could argue there's an opportunity to get the maximum potential out of everybody. If you knew how to train, not trains is kind of a funny word, but how to improve people's learning or teaching or whatever, however you want to describe it.
I think starting earlier is
not just facts, but behavioral as well. So the point I'm getting at is how much is nature or nurture here? Becomes an interesting question.
Yeah, that's an interesting question. One of the ways to answer are studies on the monozygotic twins. And there are some of these studies. I know of one which was done in England. And there was this mother who had two girls. They were monozygotic twins. And again, to explain, monozygotic twins would have identical DNA because they developed from one zygote. But not only that, they would have very, very similar epigenetics because they are grown in the same womb, they are under the same environmental factors up until birth. So not only they have identical DNA, but they have very similar environment up until birth. But then she gave them away for the adoption. And they were adopted into two different families. And then when they were about 50 years old, they were pulled into this research, which was trying to determine, like, personality-wise, how similar or different they are. And what the researcher came to is that their differences level was higher than those among general randomly selected population, which means that despite having almost identical nature, they had different nurture, and that resulted in them being totally different people. So that is one piece of information I have about that. But in response to your other part of the question about how we can improve everyone's outcome and learning, I think that the key is early start, because the earlier you start, the more time you have to master the skill. I'm very grateful for my parents, because they started me on English when I was about, I think, 5 years old. You know, I'm a kind of a product of a weird time. It was right when Putin's time was beginning, and he hadn't yet shown his true self. So it was kind of good. And during these kind of good times, there was a lot of English learning opportunities, and all of these English learning centers were opening everywhere. And one of them opened right by our house. And I think I started when I was like 5. And due to that, it helped me to learn a lot of vocabulary. Like a lot of my vocabulary still comes from early child times, when I basically was not occupied with anything. Like over time, I think it's not that when people get older, their brain functions deteriorate. I think it changes, because like we master executive function, partially because until about 33rd decade, you don't have fully formed synaptic connections in the frontal cortex, which is responsible for executive function. So executive function gets better. People make more balanced decisions. But in terms of the speed of learning, I think when the brain is not occupied yet with like emotions, responsibilities, and you can pretty much learn from the scratch. So I think the earlier you start, the better the outcomes would be.
I'd like to make a comment that we've only got technically about 15 more minutes. So let's let Valeria finish up her presentation, and then we can have questions after that.
Yeah, there's not much left. I only have 4 slides left. So, but thank you.
I just wanted to make sure, yeah.
Yeah, thank you for that. So this slide actually depicts what we just talked about, the dynamics of the axon formation. So during the postnatal brain, if like the big event of the prenatal brain development, like the biggest part of it is the forming enough neurons and forming the pathways. But then if you talk about like big postnatal event, it would be formation of more interneuronal connections, not connections between, let's say, cortical neurons and thalamic neurons, but the connections between cortical neurons. So, and here, actually, the process follows the same logic. There is an overproduction of synapses during first several months after birth. And then there is another non-pathologic regressive event, similar to the nephron mass extinction, known as synaptic elimination and stabilization, which means that those which are not eliminated get stabilized, and they become like a stable highways between the neurons. And this lasts until adolescence. And here we can see on the chart the number of synapses per 100 micron of the taken tissue in the visual, auditory, and prefrontal cortex, which are shown with red, blue, and green respectively. And these are the conceptual days. So we can see that right after birth, there is a burst of new synapses in all 3, but then once you grow, as you grow, there is steady decline in the number. And another point about myelination is that it starts prenatally, but it's not finished until the 3rd decade in the frontal cortex. And that is where our executive function, conscious thoughts are coming from. So just a bit of very basic neurochemistry part. In the developing brain, we have gamma aminobutyric acid, or GABA, which serves as a main excitatory neurotransmitter. But then shortly after birth, it switches and it becomes inhibitory transmitter. Due to the change in the ion channel structure, new of them formed, and also oxytocin release, which affects these channels. And in the adult brain, main excitatory transmitters are aspartate and glutamate. And also these two substances, noradrenaline and acetylcholine, they regulate the sleep-wakefulness cycle. And in my last two slides, so this is just about explanation of what was the picture which I placed on my Meetup frontal page. So this is a functional MRI or fMRI from the early brain, and this with red, it shows like the activated zones, and we can see that A is the primary visual cortex and B is the somatosensory and motor cortex. And how you do these studies is like you detect the activity of the brain when the person is doing certain action, and that is how you see where the activity is. And the final thing, it's kind of less to the finishing of this presentation, but more as a connection towards one of my next topics, which I wanted to discuss, is pain. So there's a lot of misinformation going around in all of these anti-abortion groups about how like 10 weeks fetus can feel pain, how they can like try to evade the whatever abortion instruments. I've seen a lot of things and some of them look completely like Photoshop. But what do we know from the actual studies? So the nociceptive reactions, which are response to pain reactions, for example, the withdrawal reflex. So they can be detected since gestational week 19. And by gestational week 20th, we can detect, yes?
So that first one, I mean, we also, as I seem like I've learned at one point, you know, jerking your hand from the hot stove doesn't even reach the brain. Your spine
No,
kind
it
of takes
doesn't.
care
It
of that,
doesn't.
right?
That
so that
is-
first
that
one you're
can-
talking about is not necessarily even involving the brain.
No,
Yeah,
it's not.
okay.
So there's another good thing to discuss, the reflectorial arches. Basically, that is like the pathway which the stimulus comes from. And you're absolutely correct to say that when you accidentally grab something hot or cold, or just something that is disgusting, like, you know, if I think that if I accidentally touch a cockroach, my brain wouldn't even notice, but my hand would be totally withdrawn. So you're absolutely right. These reflectorial signals, they are just involving the spinal cord. Like there is no need for a complicated brain involving response. These arcs are just peripheral receptor, axon, neuron in the spinal cord, interneuron in the spinal cord, motor neuron in the spinal cord, axon, and the muscle. It doesn't go upstairs because it's a simple response. So by gestational week 20th, we can detect certain substances in the umbilical blood when the needle is inserted into the abdomen. And these are things like cortisol, beta endorphin, and noradrenaline, and they are known pain responding substances. But what is interesting is that facial expressions, which are reminiscent of adults experiencing pain, they can be observed at the gestational week 28. And that is partially suggestive that that is when this pain arc starts involving the brain. And
it's after the cortical formation.
Exactly. After the thalamocortical pathway is formed, after we have cortical neurons present there, that's when we have the facial expression response involved in the pain response. So how can we study that? So there could be painful stimulations, which are done by venipuncture or heel lances, of course, in a sterile conditions, done on the preterm infants. And we can detect using this system what happens in terms of their hemodynamics. And hemodynamics stands the blood flow. And not only just hemodynamics, meaning heart rate, but we can also detect the oxygenated and deoxygenated fraction of hemoglobin and total hemoglobin, and in response to the pain. And the advantage taken here is that the transparency of biological tissue to the near infrared light That's what we can use here to detect, because like, for example, the absorption of near infrared light by oxygenated and deoxygenated hemoglobin would be different. So that is how we can detect the heart rate change, the peripheral oxygen.
That's how those little finger sensors work in the hospital, is it not?
That is a saturation. That is one thing which you can do. But these finger sensors, I think, would only read your saturation percentage. I don't know if they would, if now they're cool enough to automatically calculate like the heart rate, and it also would be in the whole body, not just in the head. Because I think here the point is that you want hemodynamic response, particularly in the somatosensory cortex. That's why the detectors are placed around where the somatosensory cortex is located, while the finger thing would tell you from the whole body. So with that, that is all I have. And now we can discuss any additional questions which might still be present. I'm going to try to return all of your faces here.
I have a question that came up towards the end. It's part of the reason I was asking the question at the beginning, about how many neurons is it between the cortex and the reaction. The fact that you jerk your hand back from the stove again makes me think there's got to be a connection in the an additional connection in the spinal cord between the spinal cord and the periphery.
Yes, and we can even count these neurons. So the reflectory arc would look like you have a receptor, let's say in the skin. Then there's an axon, which goes to the first spinal neuron. That is the second cell involved.
There we go. Okay.
These are two. So now there is variability. There are some pathways which would go next directly to the motor neuron, and that would be 3. In this very simple arc, you would have only 3 neurons, but there are more complicated arcs. They would involve what is called inhibitory neurons, which would delay your movement response. And these inhibitory neurons would also be located in the spinal cord, and then there would be 4.
Okay, interesting.
Yeah, so in this arc, you would have 3 or 4. Now we can calculate the thalamocortical pathway, or corticothalamic, no, depending on which direction you want to start. Let's say that we start from the cortex. So you have 1 neuron in the cortex. That's number 1. Then it goes to the thalamus. The nuclei of thalamus would have the 2nd neuron. Then the 3rd neuron would be in the spinal cord on the level of where this axon from thalamus goes. And then it would go to the muscle. But again, there might be more complicated ones, which after thalamus would go to other brain structures, like, for example, the hindbrain. And then there will be 4th neuron in there.
Okay.
So, Valeria, thank you for this interesting lecture.
Thank you so much.
Since you have a great deal of interest in brain activity, I'm going to suggest a topic to you just for the fun of it.
That is very welcome.
I keep on reading things about direct and AC and DC cranial excitation, external cranial excitation having some very interesting effects. So, for instance, I just read a paper using AC cranial excitation of 2 areas, of the brain, basically, improving memory function for people who are like me, who are now 60 years or older. And this seems to be a very interesting field, but it'd be wonderful with someone who's got the kind of knowledge you have, who could do some kind of a summary, an overview of what's been happening and what's going on, and does this make sense? Why does it make sense?
I could totally do that. So could you please repeat again? So what was that? It was cortical excitation?
Yeah, so what they're doing is, it's the simplest way to describe it is they basically put electrodes on your skull in various places, and they apply either direct current or they apply alternating current. The alternating current is attempting to to improve or change to some extent your oscillatory behavior, your actual brain waves, if you will, to put it this way. And the DC is just simply an excitation of various areas of the brain. And, they've been shown to do things like provide people with, in some very extreme cases, with savant-like capabilities. I just saw a paper, a reviewed paper, where it talked about where they were doing certain AC excitations, and they were showing that it improved people's performance on standardized memory tests. The kind of
That
simple
is
tests.
interesting.
The
Yes,
kind of simple
I'm
tests like
interested in learning
remember the
about
following numbers or letters or something like that. And okay. So
yeah, I'm interested in learning about that, and I could include this into our event plan.
Yeah.
So
Okay.
thank you
So
for
it's suggestion.
Yeah, that is great. We have a lot of plans. The group is going to be very active. Thank you so much. All right, so I think it's about the time, and I greatly appreciate everyone for the attention and for questions, for active participation. Thank you very much.
Thank you very much. It's very,
Yeah,
very
thank you.
interesting.
Yeah, thank you. I will, once I get the video, I will post this. If everyone agrees, we now have a YouTube channel for American Atheist and Agnostic Society, which we created. And if it is okay, I will post this in there as well.
Yes, I did mean to ask that in the beginning to make sure there's no objection from anyone to the recording and, as Valeria says, to posting on YouTube.
Yeah. Great. Thank you so
Without
much.
objection, so ordered. I will stop the recording. Thank you very much, Valeria.
Thank you.